Hello dear students, welcome to the next session of Principles of Inheritance and then the Variation. So let's discuss regarding some other terminology which are used in the genetics. The next terminology includes the homozygous and heterozygous. See, in genetic cross, very often we will be using the two terms. Whether the plant is heterozygous in nature or the homozygous in nature, or the expression of a particular character is heterozygous condition or the homozygous condition. So what this heterozygous and homozygous refers to? Let's understand that. See, during his observation and studies over the pea plant, Mendel proposed that in a true breeding, the tall or the dwarf variety of the pea plant, allelic pair of genes means one pair of genes which are slightly different from each other but coding for a particular character in contrasting manner, means slightly different from each other. So such allelic pair of genes means the pair of genes which are alleles to each other. Say for example, for a character called as height, they will be identical. Such allelic pair of genes for height are identical, then the allelic pair will be denoted by symbol capital T and capital T for the true breeding tall plant, and small t and small t for true breeding dwarf pea plant. Didn't understood? Shall I repeat once again? Okay. See, in true breeding tall pea plant or true breeding dwarf pea plant. So this sentence applies only to the true breeding tall plant and then the true breeding dwarf plant only. True breeding means so they will be true for the character what they express. Tall means it will be a purely tall, dwarf means purely dwarf without the blending or mixture of the genes. Means here, in that true breeding tall pea plant, the allelic pair of genes, they will be identical. Means for the tallness here, the two identical genes are present or alleles are present. For dwarfness, two identical genes or the alleles are present. So if same type of allelic pair is present or the allelic pair contains identical genes, then such a condition is called as homozygous. Similarly, okay, next, if an individual is having two different alleles, say for example, one is capital T and other one is the small t. But here you know that the one who is denoted by the capital letter codes for the tallness, and one which is mentioned or denoted by the small letter codes for the dwarfness. But here the plant will be tall, but it is having a pair of contrasting alleles. Are the genes which code for the contrasting traits of a character. Such a condition for a character, it is called as heterozygous. So in any individual, whether the plant or the animal, okay, if it is having two different alleles for one character, such condition is called as heterozygous. Similarly, if an individual is having identical alleles for a character, then it is called as a homozygous condition. Andre allelic pair alli ee alleles identical agidre homozygous anta kariತೀವಿ. If they are different from each other, then it is called as heterozygous. So both these terms, that is homozygous and heterozygous, was proposed or coined by the two scientists. What are those means? What are the names of those scientists means? One is Bateson and another one is Saunders. So heterozygous and then the homozygous terms were coined by Bateson and Saunders. Homozygous means it's very simple. In any individual, if the allelic pairs are identical for an character, it is homozygous condition. If the allelic pair for a character is different from each other, then it is a heterozygous condition. Clear, guys? That is regarding the heterozygous and homozygous condition. Then the other terminologies like genotype and then the phenotype. So we will be very oftenly using the genotype of the plant is heterozygous dominant or homozygous dominant. So you know that one homozygous means allelic pair will be identical. Heterozygous means allelic pair will be having a slightly different alleles of the same gene. So what this genotype and phenotype means here, genotype refers what is the genetic complement of that particular organism, whether it is homozygous dominant or heterozygous dominant. That representation of a genetic complement of an organism is called as, for a particular character, it is called as genotype. So genotype is nothing but a representation of a genetic complement of an individual organism for one or more characters. Example, for the tallness, whether it is homozygous tall or heterozygous tall, or the plant is dwarf, whether the plant is dwarf. So instead of words, we represent their condition, genetic complement, in the form of a combination of alleles. Such a representation is only called as genotype. So here capital T as a combination, capital T, small t as a combination, small t as another combination. These are called as genotype of the particular character of that organism. So what this symbolic representation denotes here means genotype of the organism for a particular character, here called as plant height. So by seeing this one, we can say that one, the plant is homozygous tall. This one, the plant is heterozygous tall. So by seeing this, we can say that one, the plant is dwarf. We know that one, capital T denotes tallness and small t denotes dwarfness. Right? So in this way, genotype is the representation of genetic complement of an organism or an individual for one or more characters. That is called genotype. Similarly, what is phenotype then? See, phenotype is nothing but observable feature of an individual or observable morphological feature of an organism is only called as phenotype. Say for example, her hairs are long, okay, he is fair, his eyes are blue, his hairs are curly. So what these examples indicate? Curly hairs, long hairs, blue eyes, okay, fair skin. So these are the observable feature of that individual. You already observe by seeing that only you can say which features are present. Yes, that organism is having only 2 legs or the appendages, or particular organism is having 4 legs. So appearance of that 4 legs is a feature, observable feature of that organism. Appearance of long hairs, that is an observable feature of an organism. She is tall, he is dwarf, means what you are representing there, what you can observe in that individual, tallness or dwarfness. So what term you used here? Tall, dwarf or short, long hairs, curly hairs. So these words denote phenotype of that individual. So here the phenotype is nothing but observable morphological feature of an organism. And remember, this phenotype is determined by different combination of alleles. Different combination of alleles. Means what character is expressed or feature is expressed in that organism, that is as a result of combination of different combination of the alleles. Example here, tall. The plant is tall or that organism is tall. So here what this tall represents? Phenotype. Similarly, dwarf. So that is also representing a phenotype. So let's clearly understand this one here. If the genetic complement of an individual of a character is expressed in terms of allelic pairs, this representation is called as genotype. See, if you write capital T together, it is homozygous tall. If you write it in words as a tall means this is phenotype. If you represent their genetic combination or genetic complement, it is genotype. See here, dwarf, it is mentioned in terms of words. So it is phenotype. But here it is written as small t as a combination of alleles. So this is genotype. Genotype means we represent it in the form of a pair or the combination of alleles. Phenotype means that we write in terms of words, what we observe in that organism. So observable feature of an organism is a phenotype. Genetic complement of that particular character of an animal or the organism is a genotype. So this is what regarding the homozygous and heterozygous, and then genotype and then the phenotype. Make a note of these points. Later we will discuss regarding some other terminology. So the next terminology or the terms are dominant and then the recessive. What do you mean by dominant and what do you mean by recessive means? So during his experiment, the Mendel, what he observed, then he proposed that when two dissimilar factors or the genes were present in a single individual or the single organism for a particular character. For one character, if two dissimilar genes or the factors or alleles were present means, so in such condition what happens, only one is able to express. Only one is able to express. Based on this, the dominant and recessive are defined. How means? So when two dissimilar factors are present for the one character in single individual means that condition is heterozygous condition. So the one, here the one is nothing but the gene. So the one that express in heterozygous condition, then that one or the gene is called as a dominant gene or the dominant factor. Understood? What do you mean by dominant means? When two dissimilar factors were present in a single individual, the one that expresses in that heterozygous condition is called as a dominant factor. Similarly, the one which fails to express itself in heterozygous condition is called as a recessive factor. Recessive factor or the recessive gene. Say for example here heterozygous condition is capital T and then the small t, means the plant is in heterozygous condition. So which one get expressed here means that is the plant will be tall even though it contains one allele coding for the dwarfness. Dwarfness ಗೆ ಕೋಡ್ ಮಾಡ್ತಕ್ಕಂಥ ಒಂದು allele ಇದ್ರೂ ಕೂಡ capital T or this allele which code for the tallness, it get expressed even in the presence of the allele coding for the dwarfness. ಅಂದರೆ ಡ್ವಾರ್ಫ್ನೆಸ್ ಮತ್ತು ಟಾಲ್ನೆಸ್ ಎರಡಿಗೂ ಕೋಡ್ ಮಾಡ್ತಕ್ಕಂಥ ಅಲೀಲ್ಸ್ ಇದಾವೆ, ಬಟ್ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಿದ್ದು ಒಂದು ಮಾತ್ರ, ದಟ್ ಈಸ್ ದ ಅಲೀಲ್ ವಿಚ್ ಕೋಡ್ ಫಾರ್ ದ ಟಾಲ್ನೆಸ್. ಸೊ ಈ ರೀತಿ ಹೆಟರೋಜೈಗಸ್ ಕಂಡೀಷನಲ್ಲಿ ಯಾವ ಜೀನ್ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗುತ್ತೋ ಅದನ್ನು ನಾವು ಡಾಮಿನೆಂಟ್ ಜೀನ್ ಅಥವಾ ಡಾಮಿನೆಂಟ್ ಫ್ಯಾಕ್ಟರ್ ಅಂತ ಕರೆದ್ವಿ. And the one which failed to express its character in heterozygous condition is called as recessive factor. So here the capital T is denoting a dominant factor or the dominant allele and small t is denoting the recessive factor. See, dominant and recessive can be applied for heterozygous condition. Remember that. Then in other words also we can say what is dominance and what is recessive means. Say that dominant allele influences expression of its phenotype. Even in the presence of its alternate allele. Even in the presence of its alternate allele or the contrasting allele. So this is what happened in this case. Even though alternate or the contrasting allele is present, but the dominant allele expresses its character, that is tallness here. Similarly, but recessive allele expresses its phenotype. When recessive allele express its phenotype only in the presence of its identical allele. See here, what example you can see? That is example, small t and small t, it get expressed as a dwarf plant. But here, dominant allele which influence the expression of its phenotype even in the presence of its alternate allele. Here example, capital T and small t, so that codes for the tallness or the tall plant. Is it clear? Even in the presence of its alternate allele, the allele get expressing its own phenotype. But here, the allele which expresses its phenotype only if the similar or the identical allele is present, then it is called as a recessive allele. So this is what the concept of a dominant and a recessive, or the dominant and recessive. Make a note of these points now. So the next one is the concept of segregation, means how the genes or the factors get segregated from parents to the offsprings. See, as we already learned regarding the monohybrid cross or the one gene inheritance, from the observation of that monohybrid cross, so what we observed in that means the recessive parental trait, say for example dwarfness. So in monohybrid cross, the cross is carried out between the pure tall variety of pea plant with the pure dwarf variety of the pea plant. That dwarfness is a recessive character. So in that cross, the parental recessive trait get expressed only in F2 generation without any blending. Means during that cross, in the F1 generation, no plant was found to be dwarf, but all were found to be the tall. When such F1 plants were allowed for the self-pollination, in F2 generation or the second filial generation, again dwarf plants are produced along with the tall plants. So as they get expressed in the F2 generation without any blending, from this we can conclude or we can infer that. So what we can infer is, when the tall and then the dwarf plant are present, when they produce the gametes by a process called as meiosis. See, we know that the production of gametes takes place by a process called as meiosis. So what happens during that meiosis means, in meiosis, the alleles of the parental pair for each character, they separate in such a way that, means the alleles of a parental pair, they separate in such a way that only one allele will be received by or transmitted to a gamete. When a gamete is produced, that gamete will receive only one allele of each allelic pair. In such a way, the segregation of the alleles takes place during the formation of gametes from the parents. Means the meiosis reduces the chromosome number to 1 half. If the one chromosome is present in the parent, means how much chromosome will be received into the gamete, means only half. If 16 chromosomes are present in parental cells, means how many chromosomes will be received to the gamete, means half of the 16, that is 8. Like that in human beings, there are 46 chromosomes are present in human beings. How many will be transmitted to the gamete, means half of that is 23. Each gamete will have 23 chromosomes. In such a way, during meiosis, for the production of the gametes, number of chromosomes will be reduced or decreased to 1 half of the number of chromosomes in parental generation. So that a gamete always receives only one chromosome for each type of the chromosome and only one factor for each character. For each character there will be a presence of 2 factors as alleles, right? But only one factor will be received by the gamete during the formation of the gamete by a process called as meiosis. Similarly, from a chromosomal pair, only one chromosome will be received by the gamete for each chromosomal pair. So let's understand this one, okay, with elaborate explanation or by taking an example. Before that, make a note of these points now. See how the segregation of alleles takes place during the meiosis process while the formation of the gametes. So segregation of alleles is a random process. It is not the controlled one and it is not the prescribed way. So it is a random process in which alleles can get segregated in any manner. But how their segregation takes place? Let's understand with example. So in such a random process of allelic segregation, there is a chance of 50% chance for each gamete to receive the either allele. So when 2 alleles are present, the gamete is having 50% chance to receive the first allele or the 50% chance to receive the second allele. Let's understand this one with examples now. Say for example, if a plant is homozygous tall, or if the plant is homozygous heterozygous tall. These are the 3 possible conditions for the plant height, right? So here the tall plant, its genotype is represented by identical pair of alleles coding for the tallness, that is capital T, capital T. When such plant produces gametes, when it produces gametes, each type of gamete produced will receive the similar alleles. Means they have to receive, here one pair of alleles are present. Remember, during segregation the gamete will receive only one of the allelic pair. So only one T is received from this for this gamete and one T is received to this another gamete. Say how many gametes are produced? 2 gametes are produced and both are similar to each other, are identical. Similarly here in dwarf plant, here also the gametes produced will receive the similar or the identical allele. So this happens when a plant is pure for its character or the trait. This is homozygous tall, here homozygous dwarf. But what happens if the plant is heterozygous tall? Means its allelic pair is having contrasting pair of genes coding for a particular character or the contrasting traits of a particular character. Then here the alleles which are received by the gametes formed, they differ from one to the another. Means one of the gamete receives a dominant trait and one of the gamete receives a recessive trait. So this gamete produce either it can receive the dominant allele or the recessive allele. There is a chance, 50% chance for receiving a dominant allele or 50% chance for receiving the recessive allele. Means here it can receive either the capital T or the small t. Similarly, this gamete also either it can receive the small t or the capital T. Always there is a 50% chance. Say for example, let's understand this with a natural or a daily life example. Say for example, in a closed container, one apple and one orange is there. One apple and one orange is there. ಸೊ ಬ್ಲೈಂಡ್ ಫೋಲ್ಡ್ ಅಂದರೆ ನೀನು ಕಣ್ಣು ಕಟ್ಟಿ ಯಾವುದಾದ್ರೂ ಒಂದನ್ನು ಮಾತ್ರ ನೀನು ಆಯ್ಕೆ ಮಾಡ್ಕೋಬೇಕು ಅಂತ ಹೇಳಿದಾಗ ಆ ಕಂಟೈನರ್ when you put your hands in that one, you will get either the apple or the orange. ಒಂದನ್ನು ಮಾತ್ರ ತೊಗೋಬೇಕು. ಅಂದರೆ ನೀನು ಆರೆಂಜ್ ಆದರೂ ತೊಗೋಬೋದು ಅಥವಾ ಆ್ಯಪಲ್ ಆದರೂ ತೊಗೋಬೋದು, ಬಟ್ ಎರಡರಲ್ಲಿ ಒಂದು ಮಾತ್ರ ತೊಗೋಬೇಕು. ಮೀನ್ಸ್ ವಾಟ್ ಈಸ್ ದ ಚಾನ್ಸ್ ಆಫ್ ಟೇಕಿಂಗ್ ದ ಆರೆಂಜ್? there is a 50% chance to take the orange, and similarly 50%, there is a 50% chance you may get the apple, right? Similarly, when one pair of alleles are present for a particular character, the gamete is having a 50% chance to receive any one of the allele or either of the allele. It may receive the dominant allele or the recessive allele. Out of two, it may receive any one allele only. So there is a 50% chance of receiving either allele. Okay. So like this, gametes are produced by meiosis process. Then such gametes are when undergoes fertilization. If these two alleles, for example, the gamete which is containing a capital T, that is the dominant allele, and from the dwarf plant, the gamete containing the a dwarf allele coding for the dwarfness. If they undergo fertilization, it is represented by cross. Then the resulting zygote, here what zygote is formed, it will be having with the combination of both parental alleles. Say for example, here it is representing the pollen and here it is representing the egg or the ovule from the female part of the plant. When they get crossed or undergo fertilization, they produce the zygote, and that zygote will have the combination of parental alleles, and here this zygote is having diploid condition, that is representing the diploid condition, and the egg is representing haploid condition, and here pollen is also representing a haploid condition, and the zygote resumes the diploid condition of the parents. So here the tall plant, it is diploid in nature, the dwarf parent plant is diploid in nature, but the gametes produced are haploid. Such haploid gametes, when they undergo the fertilization process, and the diploid condition is again resumed or reversed in zygote condition. So zygote. So this plant will develop as a heterozygous tall plant. Why? Because there is a combination of what is that one? Both type of alleles coding for the character called as plant height. So this plant will develop as heterozygous. It is called as heterozygous tall plant. It will develop as a heterozygous tall plant. So in this manner, the segregation of alleles takes place during the gamete formation. So during the segregation, always there is a 50% chance for a gamete to receive either of the allele. Is it clear, guys? So there is a formula for calculating the number or the type of gametes formed. So that is very important for the NEET examination. So formula for calculating formula for calculating number of types of gametes formed. Number of types of gametes formed or produced is, what that formula is nothing but 2 to the power of n. So thus that formula is nothing but 2 to the power of n. So let's verify whether this formula is correct or not. And what n represents here? Here the n represents number of heterozygotes. See, for example, here in this plant, for the character called as plant height, so what it is representing? Homozygous condition, means no heterozygous condition is present. If you apply the formula, how many type of gametes will be found means, so 2 power 0. 0 is representing no heterozygous are present. 2 power 0 means that is equal to 1. So only one type of gametes are produced here. Means all the type gametes will be produced having same allele, means all alleles will belong to the same type or one type only. Here also no heterozygous condition is present. So the number of types of gametes produced is 2 power 0. So only one type of gametes are produced, means all gametes will be having a small t as a allele or the dwarf allele. Similarly, if you come to the heterozygous plant here one heterozygous condition is present. So the number of types of gametes formed is 2 power 1. Why? Because here one heterozygous condition is present. 2 power 1 equal to 2. So 2 different types, means 2 types of gametes are produced here. One is having the dominant allele, another one is having the recessive allele. So here 2 different types of gametes are produced. Like this, so we can calculate the types of gametes formed by using the formula 2 power n, where the n is representing number of heterozygotes. Similarly, for example, in a plant or in an individual, if 3 heterozygous conditions are present, then how many types of gametes are produced? It's very simple. We got the value of n here. If you substitute that value of n here, means that is 2 power 3, that is equal to 8. So 8 types of gametes will be produced. Is it clear, guys? So this is what regarding process of gamete formation or the segregation of alleles during the formation of gametes by a process called as meiosis, and how to calculate the number of types of gametes are formed during the meiosis, during the formation of gametes. Is it clear, guys? This formula is very important in order to solve various type of numerical questions regarding the types of gametes formed. Okay, so this is very important for the NEET examinations. Remember it. So this is enough for today's class. And next class we are going to discuss regarding the other aspects of the genetics. Hope you have enjoyed and understood today's class. Thank you, guys.